So today we're going to continue our discussion about the retrovirus family. In the last lecture we talked about how retroviral RNA is converted from single-stranded RNA to double-stranded DNA. We're going to finish discussing that by talking about how the double-stranded DNA then becomes integrated into the host chromosome. Now, integration of the double-stranded DNA occurs relatively randomly in the host genome. There are hotspots, areas that are preferred for integration, but integration is roughly random in terms of retroviral double-stranded DNA being integrated. And once the DNA, retroviral DNA, is integrated, it's known as proviral DNA. The enzyme that's responsible for the integration is an enzyme called integrase. And what integrase does is it takes the host double-stranded DNA, makes a break, and then the double-stranded viral DNA is inserted. Now the actual process is much more complicated than that, but I don't think we need to go into that level of detail. Okay, and again, once integrated, the viral DNA is known as proviral DNA. Now, this next slide shows you once the viral DNA has been integrated into the host genome, it makes viral positive sense RNA. And that RNA that's produced does one of three things. The viral RNA can either be, can travel from the nucleus to the cytoplasm where it's translated into protein. That viral RNA can also be, exits the nucleus and forms viral genomic RNA. So some of that RNA is packaged into viral genomes. And some of the viral RNA that's produced from the integrated genome is spliced in the nucleus to form the ONV gene. That spliced RNA then exits the nucleus and is translated into protein. And that's shown on this slide. So you can see the viral DNA, the proviral DNA integrated, makes RNA. Some of the RNA is translated into protein. Some of the RNA is used to make viral genomes, packaged into virions. Some of the RNA is spliced, which is used to make the envelope protein, the surface protein on an HIV particle. Now, it's important to realize that the mRNA that's produced from the integrated copy of the viral genome is not the full-length copy of the DNA that got integrated into the host genome. So as you can see from this image here, it doesn't go from one end of the LTR to the other end of the LTR. In fact, it begins within the LTR and ends within the LTR. And essentially, that's why we have the truncated version or the smaller version of the viral RNA that enters into the cell. So the viral RNA that enters into the cell, so if you'll recall, the viral RNA that enters into the cell goes from RU5 at one end to U3R at the other end of the genome. So it doesn't contain the complete LTR sequence. The LTRs are added during the conversion of single-stranded RNA to double-stranded DNA. And you can see that here. The final product contains the LTR with some additional sequences added to each end of the viral genome to create the LTR. But in fact, the viral RNA that's actually produced from the integrated DNA is only from this R sequence to the end of the other R sequence, so essentially making these genomic copies. And that can be seen on this slide again, where you're beginning your synthesis towards the repeat U5 region, and down here the U3R region. So what's being produced here then are genomic copies of the RNA, which can also be translated into proteins. But that genomic copy doesn't contain the full LTR sequence. It's the same sequence that we saw of the single-stranded piece of RNA that infected cells and that was converted into double-stranded DNA. So that gives you a rough idea of the basics of HIV replication. So what I'd like to do now is to talk about antiviral drugs. And as we've already discussed, there is no cure for HIV. Once you become infected, the viral DNA integrates into your host cell DNA. And so unless you kill all the cells that are HIV positive, you can't get rid of that DNA. There's also no vaccine currently available to treat HIV infections, and so the only thing that we have are antiviral drugs. So I'll give you a brief history of antiviral drugs and then talk about how they relate to an HIV infection. So in the early 1900s, Paul Ehrlich came up with the idea of the magic bullet. And sort of the idea came about when looking at stains that were staining prokaryotic bacteria, but not staining eukaryotic cells. Ehrlich thought, well, what if we could put something toxic, attach that to this dye? That dye would go to the prokaryotic cell and leave the eukaryotic cell alone. So we could potentially kill the prokaryotic cell. So the idea then evolved, can we develop something that would kill these prokaryotic cells? And the challenge is to find something that kills the microbe without hurting the host. Vaccines are effective if given prior to exposure to an infectious agent, but oftentimes a vaccine is either not available, or if you become infected by a microbe, vaccines may offer little protection. And so that's where antivirals come in. They fill in the gap between where we have vaccines developed and where we don't. Although there's been a lot of research to develop antiviral drugs, in fact, there's only about 30 or so antiviral drugs currently on the market. And you may think about your own personal experience. If you think back, have you ever been treated with antibiotics? And antibiotics are used to treat bacterial infections. And probably most people can think of examples in their personal life where they've been treated with antibiotics at some point. So if you think about your own experiences, most people have been treated at one point or another with antibiotics. So if you think back, you've probably been treated for bacterial infections with antibiotics. However, if you think again about your own particular situation, most people probably haven't been treated with antiviral drugs. So why do we have so many of these antibiotics that are effective against bacteria and prokaryotes, and why do we have so few antiviral drugs? Well, there are two sort of explanations why there's not a lot of antiviral drugs. The first is that bacteria and prokaryotes are fully functioning life forms. So they contain their own transcription and translation machinery. And although they contain transcription and translation machinery, they have characteristics about them that are unique to prokaryotic cells that aren't present in eukaryotic cells. So they have a different composition to their cell wall or cell membranes. Although they have transcription and translation machinery, the proteins that do that are divergent enough that you can specifically target transcription and translation machinery of prokaryotes and not affect eukaryotic cells. So because prokaryotes are different enough from eukaryotes, you can target unique aspects of those organisms using antibiotics and not harm the eukaryotic cell, which is what you want to do when you develop an antibiotic or an antiviral agent. The challenge with viruses, though, is that they grow within eukaryotic cells and are dependent on much of the eukaryotic machinery to carry out their replication. What does that mean? Well, it means that you have very limited targets for viruses to inhibit their replication cycle. We've seen with some of the negative sense RNA viruses that some of these viruses only encode 5 proteins. So you really have a limited number of targets, and since viruses are dependent on the host cell to carry out their replication, again, you need to target specifically those viral components without targeting the eukaryotic components and obviously damaged cells. So it's much more challenging to develop antiviral agents than it is for antibiotics because there's a lot more targets and a lot more differences between prokaryotic and eukaryotic cells. The second challenge is that antiviral drugs have to be very efficient. So you need close to 100% efficiency of the antiviral drug inhibiting the viral replication. Otherwise, you'll get emergence of mutants that are resistant to that antiviral agent. So something that's 99% efficient is not good enough to control a viral infection. So when a single viral particle infects a cell, it can make thousands of viral progeny that emerge from that cell. So if you're 99% effective, that means 10, 20, 30 viral particles could emerge from that cell, even under selective pressure of the antiviral. And as these viruses emerge, they may gain resistance to that particular antiviral drug. So antiviral drugs have to be very effective. Some of the other challenges in creating antiviral drugs is that in many cases, viruses either don't grow in cell culture, so things like hepatitis B, hepatitis C, and papillomavirus, or we have no animal model for these viruses, things like measles and hepatitis C virus. So it really limits your ability to screen for these antiviral compounds and then to take these potential antiviral compounds and test them in animals to see if they'll work. So as you can well imagine, with the success of antibiotic therapy to treat bacterial infections in the 1940s and 1950s, there was a great deal of enthusiasm for the creation of antiviral drugs. And scientists initially and companies initially assumed that you'd be able to develop these antiviral drugs just as easily and effectively as these antibiotics were developed. Through the 1950s, though, the only antiviral drug that was developed was a sulfonamide antibiotic, which was used to treat smallpox. And another challenge associated with creating these antiviral drugs is simply the cost. It can cost anywhere from $100 to $500 million to bring a single antiviral drug to market. So one of the few success stories through the 1960s and 1970s, when there were these large screening programs by pharmaceutical companies to identify new antiviral compounds, was a drug called acyclovir. And acyclovir is designed to treat herpes simplex virus infections. So HSV type 1 causes cold sores, HSV type 2 causes genital herpes. In 1974, acyclovir was discovered, and it can be delivered either through topical cream, ingestion, or in capsule form. So how does acyclovir function? Well, acyclovir is sort of a classic antiviral compound. So I'll spend a little bit of time discussing how that particular drug works, because it really goes to show how antiviral drugs generally need to function. So acyclovir is known as a chain terminator. And what does that mean? Well, if we look at ribonucleosides, we have a base here, and you can see that it contains two hydroxyl groups, in particular a hydroxyl group on the 3' carbon. Nucleotides in DNA, deoxynucleosides, contain a single hydroxyl group on the 3' carbon. Chain terminators lack a hydroxyl group on the 3' carbon. Why is that important? Well, during DNA synthesis, the incoming nucleotide, here, the nucleotide triphosphate, interacts with the hydroxyl group on the growing chain of DNA to form a phosphodiester bond. Without the hydroxyl group on this growing chain, no new nucleotides can be added. So here's the structure of acyclovir, and this is the structure of the guanosine nucleotide. And what you can see is that the structures are remarkably the same, except for this carbon ring here. In particular, there's no hydroxyl group here where the 3' carbon would be. So this drug lacks the 3' carbon and the hydroxyl group attached to it. So if this nucleoside, this acyclovir, was to be inserted into DNA, and it could base pair with cytosine, which is what G normally binds to. If this nucleotide were to base pair with C and become integrated into a piece of DNA, there's no hydroxyl group to continue extension of the DNA chain, so DNA synthesis would stop.